we've been talking about DNA and how awesome DNA is, but what we have not talked about is how DNA actually codes for your traits. Say, for example, we're talking about your eye color. Yes, the DNA codes for the genetic information that codes for the color of your eyes, but how exactly does that work? Well, keep in mind that your eyes are the color that they are based on a pigment that is inside the eyes. And in order to have that pigment, your DNA must code for proteins that help make that pigment, which gives them their color. So today what we're going to talk about is how your DNA leads to a protein. And this is actually called protein synthesis. Just like it sounds, whenever you say synthesis, that means to make something. So protein synthesis means to make protein. Whenever we're talking about protein synthesis, we also need to realize that this is happening in all of your cells, okay? Because all of your cells contain your DNA. And while not all of the DNA is turned on at any given time, it's based on what type of cell we're talking about, that DNA is present in the nucleus of all of your cells. What we have to consider is, how are we going to get the genetic information that's in the DNA, which is inside the nucleus, out of the nucleus so that the cell can start producing the proteins that it needs to make based on the genetic instructions?
Well, in order to answer that question, we need to go into the process of protein synthesis.
In protein synthesis, there are two major steps, something called transcription and then something called translation. There's actually a lot of trans words that we'll get into in biology, but just stay with me here. Transcription has a C in it, and translation has an L in it. And I remember that C comes before L in the alphabet, and so that helps remember that transcription comes first. I don't know, that's always kind of helped me. Now, transcription is when we're going to transcribe the DNA into a message. And if you think about that, think where DNA is. Well, DNA is always in the nucleus. So we're starting transcription in the nucleus. And what we're going to do is the DNA is going to have matching RNA bases matched up to it with the help of some enzymes to make mRNA. So in transcription, we make mRNA. We make a message out of RNA. So at the end of transcription, we have made an mRNA molecule. Remember, that's messenger RNA. And one great thing about being an mRNA is you can get out of the nucleus. So the mRNA goes out of the nucleus where it's going to attach to a ribosome. Remember, ribosomes make protein. Also remember that the R for ribosomal RNA, that's what makes up a ribosome. So the messenger RNA is going to go to a ribosome, which is made out of rRNA. And now what we're going to do is we're going to use the message to build our protein. This step is going to be called translation because we're translating our message into protein. And we are also outside of the nucleus. We are in a ribosome, so that's where our location is now. You know, you can find some really great clips, some animations on translation that are just fantastic. Really, what I like to think of is you've got this mRNA and it's in a ribosome, and then you have these tRNAs coming in. tRNA, remember that stands for transfer RNA. Well, their job is they have an amino acid on them. And an amino acid, if you remember, is the monomer for a protein. So what that means, it's a building block for protein. So if you get a bunch of amino acids together, you build this big old protein.
All of these tRNAs are looking for the matching bases on the mRNA, and when they match up, they will drop off the amino acid that they are carrying.
So eventually what ends up happening is you get this big chain of amino acids that have to match up with the coding on the mRNA. The different amino acids are held together by something called a peptide bond. It's basically a protein bond, and you end up at the very end with a really long protein made of all these amino acids, which now can be used for all kinds of different body processes. And that could be something, for example, if we're going to make eye pigment. One important thing to mention is that when the tRNA is bringing in the amino acid, it reads the letters on the mRNA in threes, meaning every time you have 3 bases, that's how it reads it. So it doesn't read one letter at a time, it reads it in triplets, in groups of 3. That's called a codon.
A codon is made of 3 bases.
So, for example, if you had an mRNA strand, it wouldn't just read A, and then U, and then G, and then some other letter. It would read AUG together, a triplet, a codon. And so because it reads in 3s, when we get into mutations, we'll talk about how if you end up taking out one letter, that could be very bad. Because if you read in 3s and you take out one letter, that will mess up your reading frame for the rest of that strand. So when we get to mutations, we'll really go into that. One thing I'd really like to encourage, with topics like this in biology, it's really great to have some kind of activity so that you can really walk through it. Because it's really hard to visualize this protein synthesis, but if you can do an activity where you're actually involved in it, it will help it make a lot more sense, I promise. So if you do a Google search for PBS DNA Workshop, just all of that, you will pull up this great resource. The PBS DNA Workshop not only has a little workshop of DNA replication, but it also has a great protein synthesis activity where it will actually let you do the protein synthesis part, both the transcription and the translation. Just highly recommend it. Anyway, that's it for Amoeba Sisters, and we remind you to stay curious.